Yeah.
Hey, welcome.
Hi, good morning.
Good morning. How's everything going?
Good, good. How are you?
Doing well. Doing well. All right. We got a couple people joining. I don't know. Do some pre-festive I don't know, conversations ahead of July 4th stuff happening. I'll be spending the afternoon with some of my family with the kids. We're gonna go do something with them, and then we'll go join some friends for the evening. I don't know. You guys have any plans?
I mean, I have to do some work. I have a deadline so I work at a small pharmaceutical company called Plus Therapeutics. And, with many small companies, you just work a lot of overtime. And so I have to, yeah, unfortunately, I have to work because I have a deadline. But we're gonna go watch the fireworks in San Diego tonight. So, yeah.
Glad to hear that. You'll at least get a little bit of a respite.
Yeah,
Go do some socializing anyway.
yeah.
Here, and I don't know. Let me just see. I want to just make sure that I can. Here we go. All right. So, anybody have any specific questions? I mean, how's the week gone, so far? I mean, this is just sort of open time for, anything you guys want to talk about. I'll hit on, a little bit of, sort of my perspectives on some things this week, and just try to, give you guys some clarity. But, hopefully most of the things that we've gone through are pretty, straightforward and self-explanatory, but I don't know. It'll get a little bit more complicated maybe starting next week, where office hours is maybe a little bit more important. But, at the moment, it's just, if you got any questions, if there's any way that I can be, helpful, to you as we're going through the course, please don't hesitate to let me know.
I mean, I don't have any questions on the course. I think it's really fantastic so far. Um, I started the program in September, so Jennifer was doing the first two sessions that were outstanding. And
Good.
then what you're presenting is also great. I really enjoy the discussions and familiarizing myself with the FDA, different databases and websites, because I don't, look at it every day. But I think I had some questions on what I had messaged you.
Yes.
Yeah, so when you talked about the pre-submission in last week's course, you mentioned you want to time it right. And then
Yep.
I asked some questions about that. I also and this is because our company purchased an essay that they want to take through the in vitro diagnostics route.
Yep.
And, they're working with some consultants, and I'm not very closely involved. But from what I had asked you, you mentioned when you apply for a de novo, that can also, after you've completed that, you can also then market the device. Could you, because I was
Yeah.
thinking we do the pre-sub, then the de novo request, and then a 510K, which I think this essay might be given the intended use. But when you line up the timelines, that takes quite a long time.
Yeah, so the de novo is basically a way to put a product on the market for the first time. So it's like it's a way to classify as a 510K product, a product where there is no predicate. So that's the difference between a 510K and a de novo is think of a de novo as a predicate less 510K. And so there are a couple other steps involved there. FDA has to do some additional, work in classifying, creating a classification, setting up a product code. You can actually get a product code for a device within an existing classification, not through the de novo process, but within the 510K process. So if you have a predicate that's one class, that's one product code. FDA can create a new product code within a 510(k) to designate some difference that they believe doesn't require you to go do a de novo or something. So
Mm,
one example of this is, for hemodialysis, there's one product code for a daytime, sort of regular hemodialysis machine, which has been done, for ages. Nocturnal has also been done, more or less like, for forever, but not necessarily on label. And so when the first nocturnal use 510(k)s were coming through, there were a bunch of extra accessories and things that the companies were, putting together to be able to do that safely. It got a different product code as a differentiation, and that kind of thing happens,
mm,
across, different areas. One product code where there's a whole bunch of stuff under it is KNT, and this is in the gastroenterology space. And
mm,
FDA did a big effort setting up new product codes to try to break that product code into, more,
mm,
still mammoth, but, sort of more bite-sized pieces, because KNT is sort of like a meaninglessly large, product classification. And so at
mm,
some point FDA was trying to, sort of break it out a little bit more so that, people could, make more sense of it, or so that FDA at least could, figure out how to better, sort of figure out navigating predicates and what's a good predicate, et cetera. Because just because something falls into KNT doesn't mean that it's like your device at all. Like this is
mm,
a really big bucket, right? And so the nice thing about product codes is usually you can look at that and say, oh, well, here's one product code. I'm making a similar device. It's in the same product code. This is like at least probably a reasonable predicate. I mean, there might be reasons that one versus another device is a better predicate for what you're trying to do. But with KNT, it's just, it's just too many devices, too many different types of, products. And I think it's still a little bit of a mess, but FDA did a big effort to, to try to clean that up a little bit a number of years ago.
mm.
But
In-
anyway, back to, like de novo and sort of that timeline. I mean, and with the pre-submission, I mean, you're looking at, generally FDA will try to schedule meetings these days within 60 to 75 days of your meeting request arriving at FDA. Um, you're right in that ballpark of 60, probably on the near side. And so generally I'd recommend that you propose a couple, dates right between, let's say, day 55 and day 60. And then FDA will still probably come back and tell you, well, we really don't like your dates. We think, day 65 to 75, and they'll propose a couple things, do something, so they'll give you some, proposed dates that work into their calendar. But anyway, I've historically tried to be a little bit more aggressive with that, where I'm proposing dates that are 45 to 60 days out. But invariably these days, whenever I propose those dates, FDA comes back with new dates that are further out. Wasn't always the case, but currently seems to be the situation.
uh, and during the pre-submission, can you ask the FDA for guidance? So, because you actually mentioned this in your first, in the last week's lectures, like with minutes, the recommendation is, the sponsor takes the minutes, or,
Yes.
is that the same with um, proposing the regulatory pathway, or could you ask an open question and ask, FDA, what do you think we should do? Like, how do you ask the FDA for questions?
Yeah, I mean, you can always ask the FDA what to do, but I wouldn't.
Okay.
It's best practice um is to really propose something to FDA, and then, um and then figure out what you want to do from there. You
Yeah.
generally don't want to ask FDA what you should do. Um One, because they're probably going to propose something that's not favorable to you, and you might actually have an idea of what's favorable for you.
Yeah.
Um, and if you don't know what's favorable for you, you should probably think about it a little longer before asking FDA. I mean,
Mm-hmm.
sometimes you really just want to know what FDA, thinks about something in particular. But generally, your best practice is to propose something and then and then figure out from there. And FDA will, sort of give you an up or a down or things they don't like about your, proposal. You're more likely to get constructive feedback the more detailed you go with, sort of your proposal.
Yeah.
If you just give FDA a very general set of information and then ask a very general question, they'll give you a very general answer, and that's not going to be as helpful for you. Um, so I'm trying to think now too. So for do you know a product code that's similar? Are you willing to share like a anything like so from an IVD standpoint, you know, is there a particular uh um substance that's being detected that's important, et
Yeah.
cetera? Like, because oftentimes like you can do a search of the product code database, and or you can go do a search within Orca, which will give you like better information. But like I'm generally gonna be doing like a Google search isn't bad, or doing certain product code searches and trying a couple different variations. You can usually find some things if there's an existing product code. And then oftentimes, if there is an existing product code for this type of a product, then you kind of already know what your regulatory landscape is. There was a company I was working with not long ago with an IVD product, actually with a couple in a similar kind of a scenario, but with one in particular, they thought it was going to be a de novo product
Mm-hmm.
initially,
Mm-hmm.
and we were able to identify that for this particular substance, it was they were measuring the free versus bound
Mm-hmm.
uh, sort of concentration of a particular drug. But the way that the product code was set up, it wasn't specific. And so we had a series of conversations with FDA and actually got confirmation that as long as they can demonstrate that the performance of their assay is good enough,
Mm-hmm.
that it wouldn't be a de novo. The only scenario in which it would get pushed into a de novo for that instance is if you had, let's say a less, you know, robust, assay where maybe
Yeah.
you're not as good as the predicate, but you're able to show, like in the de novo, that okay, it's not as good as the predicate, sort of sensitivity and specificity,
Mm-hmm.
but we're identifying a the free version, which isn't currently you
Yeah.
know a
Yeah.
thing, and so then you would go down this benefit risk de novo kind of discussion. But as long as they were able to to demonstrate that the measurement was was accurate enough, and there were some caveats around all
Mm-hmm.
that, and a lot of it comes to clinical outcomes.
Yeah.
So, or at least some surrogate for clinical
Mm-hmm.
outcomes. So it really depends on the situation you find yourself in. But if there's a potential product code that exists now,
Mm-hmm.
just because your product is a little bit different than the products that are on the market doesn't mean you should be de novo. There might be reimbursement reasons to go that route. Um, but nonetheless, like I would generally advise, unless there's a really major reimbursement challenge for you, you want to pick the 510(k) pathway, not the de novo pathway, because it doesn't actually save you a whole lot. It might and it even puts you in a position where maybe your path to getting reimbursement coverage is longer. And then on top of that, you still have the uncertainty about, are you able to get the reimbursement that you need still, for all that. Whereas if you're able to identify an existing product, you know what the reimbursement situation looks like, and then you can make a business decision about, does it make sense to do this?
Wow,
I
this
don't know.
is so fascinating. There were other people on the call too. I don't want to
Yeah,
totally hijack this meeting, but yeah, what I what I can share is the analyte are circulating tumor cells, but it's not in the blood, it's in a
okay.
different substance.
Like
But
in
what is
plasma or something?
no, it's a different fluid in
Okay.
the brain. But what is
Okay.
interesting here, the test we always look at, which is a 510K clear test, is cell search. But that test has a negative reimbursement decision by CMS because I mean this is, I think that was decided 10 years ago. Um, so yeah, we'll have to look into this because that makes me think going for, because it could be considered a predicate device, I guess. I don't know, but yeah, I think that's kind of, we have to be strategic there because of the reimbursement implications.
Yeah, so it looks like, here, let me just, so, and I don't have this up on the screen yet, but if I find something useful, I'll drop it in, but otherwise I don't want to like clog it up. But looks like it's product code QSA for that, or here, no, different, something else maybe. Search. Do you know what was the name of that product you said?
Cell search. Yeah,
Cell search.
yeah, I can put it in the chat.
Okay, cell search circulating tumor cell
Yeah,
kit. Yeah, so if I look, we've got basically 7 510Ks that they got. If I look here, so this one's NQI 6020. No, 611. Interesting. Here, let me go over to these. FDA ECFR is another place to go. I like the here, let me just look for 866. We're gonna go under medical devices. We're gonna go look at immunology and microbiology devices. So I'll drop this in the chat actually, if you want to go look at this.
okay.
But so when we look at diagnostic, okay, what are these? These are 6020 and 6110. Is that right? So I don't know why 6110. Looks like this one might be classified under a de novo or something. So if I go over here and I look, Yeah, QSA. So there is a de novo. There's at least one, and you might find others. For a circulating tumor cell enrichment device. But again, this is also from peripheral blood. So, what is the regulation? Yeah, from peripheral blood of patients. So it's specific in the product classification that it's blood. Probably you end up going de novo if you're collecting it from other fluid. I actually think with some of these things, FDA should be more generic in how they identify sort of the product code to begin with. But regardless, I would look into this one as well. And presumably they're already aware of it and have looked at it. I don't know if that's applicable at all. But then when I look here, there's a summary. Forgive me. There's a summary and a decision summary here. This is the most recent one, and you can see there's a whole history of them for the
Oh yeah, that's right. Yeah, Passortix. Yeah, they got that. Yeah,
so
in
that's a more
2008.
recent one.
Yeah,
But I don't know. I mean, it looks like probably you're in that de novo territory. It's hard to know if there's maybe something that I'm missing just on a quick search, but and it depends on uh, next generation sequencing, tumor profiling test. Are you doing next generation sequencing?
no, we're just doing cell characterization. Yeah, and counts.
Cell characterization and counts?
Yeah, so
I don't
we
know. Is that even a diagnostic?
so the diagnostic is actually the qualitative results of detected versus not detected.
Okay.
Yeah, and
So it's assessing that based on like a photographic image or something of the collected
yeah,
cells?
yeah,
Hmm. I don't know. It's so that's more sort of generic cell. This is I was just looking at one of these cell. Which one was that cell? It's like abnormal. Automated cell counters. You almost fall into but again, this is for blood and you're taking it from other tissues. So you're probably in that de novo sort of territory, but you almost fall into this. I just look subpart F 864. It's like hematology pathology devices, but you're not a hematology product, right?
correct.
Yeah. Well,
Well, yeah, I'll
you know
check.
what? Uh, are you identifying formed elements of the blood that are basically taken from other tissues though?
No.
Because if you look at this, So you potentially almost fall into um, an automated differential
Hmm.
cell counter classification. But anyway,
Hmm.
just
Interesting. Or other
just sort of an
bodily fluids. Yeah, okay, that's interesting. Yeah.
but this is specifically looking at hematopoietic cells
Huh.
of the blood, bone marrow, or other body fluids. So depending
Yeah.
on what exactly you're looking at, this is applicable.
Yeah.
But
Oh, that's great information.
but again, this is where like in that discussion with FDA, you'd almost want to present, here's what we're doing. We think that we fit under an automated differential cell counter. And you might actually find some predicates under 864 5220 that do exactly or do not necessarily exactly, but do very similarly, you know, or do very similar things to what you're trying to do. So I generally try to push for identifying products that are similar, even if you're de novo, you want to kind of have a sense of what are the similar regulated products out there, and you have some sense of of that already. But just trying to look at what else is there, and whether some of this would help to inform at least the testing you would do under a de novo, inform the types of studies that you would conduct, and really help narrow, sort of how it is that you would approach this. And the better armed you are with some of that data on what FDA has allowed in
Yeah.
some of the similar types of product, categories, the better off you're going to be in terms of, putting together a testing strategy and then put that in front of FDA. And then you get their feedback on your testing strategy. It's
Yeah.
like, oh, here's how we plan to test it. You might even have a protocol ready, though, maybe not, that far developed, but you want to be able to put that kind of information in front of FDA and say, hey, we're following I don't know, whatever these tests are for automated differential cell counters. You can take a look at, you know, the sample sizes for similar types of, products that are out there. I mean, I'm more familiar with, I think it's the automated cell counters versus the automated differential cell counters, but nonetheless, like or automated cell locating device is this one. I mean, anyway, some of these products in 864 might be interesting to look at, even though the hematology and pathology, they're hematology and pathology devices. So sort of you're sort of in that pathology
Yeah.
spectrum.
Yeah. That's great. Yeah, awesome. I'll stay on, but I think maybe others should ask some questions now. But that's
So,
fantastic feedback. Yeah.
but it gives you a sense. So like, it's sort of helpful here, and I'll actually share screen two. Okay. Um, so this is sort of, the process of, sort of going through and um, finding some of this stuff. You just
Yeah.
you poke around. Okay, you're over in 866. You go over to the CFR. You do a little bit of searching. You find, well, no, okay. And some of it's helpful to talk with somebody like me who's looked at a whole bunch of these kinds of things in the past. Like, this sounds like an automated cell counter, but so I go over to 864, 5200, but then you go, well, I don't know what else is over here. Oh, well, this one's actually maybe closer. So
Mm-hmm.
this kind of a thing where um you just chat with somebody who kind of knows what they're doing. It's well worth the the time and and energy typically.
Yeah.
And you'll be surprised how many people will be happy to share and help you. And there's some limitation in terms of what all you can share for the purposes of non-disclosure agreements and
Mm-hmm.
all of that. I
Mm-hmm.
mean, but nonetheless, it can help you better narrow in on some of this, even if you're able to talk about it in generalities.
Yeah.
And you can sometimes get in this case um you've got me you can throw questions at.
Mm-hmm.
Other folks on the call, I don't know, is there some other interesting thing that we can be talking about in terms of like thinking about regulatory assessments and classifications and these kinds of things that might be narrower, sort of closer to something that you're working on. Otherwise, I've got another example I can jump into either now, or I'm going to do a follow-up call on LinkedIn here in the following hour, and I'm going to go through an example there if we don't get to it here, depending on what questions you have. But it's mostly your time and looking to sort of get a sense from you all what what you want to what you want to talk about and what might be useful for you.
Hi, actually, I do have a kind of a follow-up question from what you mentioned
Sure.
before about there's a possibility that they would, uh, even from a 510(k), they would have create a new code for a new product because it doesn't exactly fit into what's existing right now. What kind
Yes.
of process would they go through, in order for that to
It's
happen?
That's just internal FDA processes. Like you probably don't even get involved in that, except that you find out that they issued you a new product code.
Got it.
You could maybe be proactive about some of that, but I wouldn't bother. I would mostly just try to focus on um, how it's similar to existing predicate products on the market. And then FDA will figure out whether they want to leave it in the existing product code, if they want to create a new product code. FDA's moved to being a little more conservative about this, where if they're going to create a new product code, they're more likely to encourage you to do a de novo. But this
Mhm.
is where it's helpful to go back to the classification itself. You don't have to. So sometimes there's a difference between the here. So if I share, I wonder if um, here there are some here. Let me product classification, new search. I'm gonna see if I can with this one. I'm gonna go back 8645220. So here I'm gonna share screen 2, and then what we'll do is we'll search for 864. What is 5220? Sorry, 5220. And here we see there are a number of different. Um, so we can see. I don't know if you can see my
Yes.
screen. Be both of these sort of side by side. It might be a little small. Sorry about that, but um, you can see. Okay, here's the language within the classification. This is the thing that's important. FDA has 9 different results. They have, semen analysis device. Well, that's not from blood. That's not abnormal hematopoietic
Mm-hmm.
cells, as far as I can.
Ah,
Yeah, like I don't think about it in that sort of way. But they classified it within this classification, and boom, you've got an automated differential cell counter. That's the sort of broad category. This language is a little bit more specific to hematopoietic cells, but FDA decided it's close enough as a predicate for some of these other ones, and they put it under this umbrella. So this is the kind of thing. So you can sometimes come in here and find out when they did that. So you can see here, 2002, they created this subcategory, right? Within, well, actually, so sorry, no, that's not where they created the subcategory. Let me see if I can find it in the, that's when they created the sort of the broader product code is this date. Let me see if I can find okay, I just got to go in to this and see if we have any information. There's nothing on sort of timing here, but we can tell if we go and we do so if we do a 510K search, so back to search results, we can do a new search. So now we can look for product code POV, and that'll give us a sense of when this, copy paste doesn't always work very well in these systems, just FYI. So let's see, let's make it 50. And we can see, actually, these go all the way back to, 1988.
Oh.
Okay, so this is actually fairly, this is fairly old.
Oh.
So for whatever reason, there looks like there were some changes made in 2002 affecting this, sort of automated differential cell counter. I don't know exactly what happened there versus, these products that were already identified on the market. And here you can see Riotech, laboratories, diagnostic screening, test kits for males from 2001. But yeah, you can see this goes, these analysis kits go all the way back into the late 80s, which was not long after the the medical device, amendments got passed. This is, we're talking, I mean, it's but like what, 35 years now, more or less, slightly longer. But anyway, so, okay, so we can go back to, this and see, okay, so this is one of those products. You see lymphocyte markers, DNA probe, and then sort of the more general differential cell counter. Okay, so the differential cell counter, GKZ. So we'll go over here and we'll do a 510K search for GKZ, right? And then you can see within GKZ what's out there. And here you have 413 results, right? So you can show them all on one page if we wanted, but anyway, and then go all the way down. And yeah, these go back all the way to 1976.
Oh.
So, and it started with hematopoietic cells, it looks like, maturity profile, assessments and things. But then, within about 10 years, FDA, instead of creating a new classification or even modifying this one to make it more broad, they rolled in, semen analysis, for instance. So just be aware of that. Like, just because this isn't a broad enough set of even if the language here is not as broad as you think it ought to be for the product that you're trying to to get on the market, that doesn't mean that you couldn't use that product classification, because there's a whole lot of regulatory precedents that might mean that product is already on the market, or you already have a predicate that you wouldn't think would fit within that classification, but that FDA almost 4 decades ago decided it's close enough, and they have a whole host of products that are on the market within.
I see. Okay. Well, that's interesting.
Yeah.
Thank you.
Yeah, absolutely. Any other, like, I don't know, we've got another 15 minutes or so till I probably sort of convert over to to another meeting here. But what so tell me a little bit about your thoughts on the discussion. There was a whole bunch of stuff I saw on, sort of the Chevron cases and different things that people were talking about. Anything particularly interesting from discussion or other things that we want to talk about?
Hi, I have a question. So earlier in the class, you mentioned regarding the submissions, where FDA, when we ask the suggestion about for FDA, it's not like, so that's what like you said, regarding the suggestion, it's not like maybe sometimes in our favor, maybe sometimes it's not in our favor. But my question is, how about the pre-submissions? So there is something like called pre-submissions, where we can write a formal written request to FDA, and they will help us and guide us to improve the product development. How is it like comparable, or when it is done? So my question is about the pre-submissions. So is it really be helpful or not?
Yeah, so if you frame your questions and you're proposing what it is that you want to do, I mean, pre-submissions are very helpful. You might want to even do an informational meeting instead if you're very early on. So, I mean, this is the kind of thing that I generally advise, clients who want to engage with FDA, but they're not necessarily at the point where they have questions to really be asking FDA, or they don't have a protocol for FDA to review yet. Is that you can just present to FDA what it is that you're trying to develop, what it is that you're working on, how you're thinking about this, without any questions at all. Or you can do a set of questions to FDA in sort of a simpler, pre-submission, but it's going to be less useful for you generally. It is helpful to have these, multiple interactions with FDA to make them familiar with what it is you're doing. But you also want to make them the best use of that time and that interaction with FDA to be able to get constructive feedback on what it is that you're actually trying to do. So if you have a protocol that you're looking for feedback on, that can be helpful to present to FDA. I've worked with some academic, organizations, and sometimes, sometimes less is more. So just because you have a set of documents to share doesn't mean you should just include it in the package and send it off to FDA. You want to be a little bit judicious about, what you include in your meeting package. And even if FDA asks for certain things, you don't necessarily have to, comply and give them this information in a pre-submission. It's not a pre-market submission as, trying to get a product on the market. Once you're at that stage, you have to navigate that a little bit more carefully. But I've had FDA ask for, can you give me the protocol for this, that, or the other thing? And, it's helpful when you have a consultant like me, working with you, because I can say, oh, well, let me ask the client. And, it's you have this intermediary where FDA doesn't think that the client is just clueless. Like they don't know exactly who to blame for not getting the document that they want. It's like having an accountant interacting with the IRS. It's sometimes just a better idea, you know, as opposed to, oh, well, I can't find don't tell them you can't find the protocol. I tell FDA, oh, well, I've asked the client about this, and I'll get back to you when I have something. And the deadline for when FDA wants something in a pre-submission passes, and okay, they'll just review what they have and give us feedback. And then, and there might be reasons for that. Like, if the protocol isn't very clean, in terms of some of the studies that were performed. Maybe I want to do a little bit of work on cleaning that up in a later submission, even though those are like the approved protocols that maybe animal testing or something was done under. You can better clean it up in a submission as opposed to just give FDA the, sort of the uncleaned up version of everything without any kind of a, basically handing them all of the documents without a map. Like what you can put together in the submission is sort of a better overview. And even though the underlying documents don't change, you can better present that as like a summary of what was done in terms of the protocol. Because, I had the one animal study, for instance, where there were 4 different amendments. Some of them were from the testing laboratory. Some of them were because the sponsor wanted, new information. But the underlying protocol itself didn't get updated, right? It's just you had this series of amendments that were put in place, and over like 2 years, and then the study was conducted. And so sometimes, that may or may not be information you want to put in front of FDA, right? Because it doesn't necessarily put you in the best light for those early conversations. You can maybe, deal with that later if you have to. Or, try to clean it up a little bit and then give it to FDA, give them sort of an overview, a summary of the protocol or something like that. But anyway, we gave them some data to look at, and then they were asking for a protocol. And, it's just, these kinds of little nuances of what you might deal with. You really want to try to make sure that you put your best foot forward in terms of your interactions with FDA. I don't know if that helps as like sort of one example. There are a ton, maybe better examples I could use, but.
Thank you so much for the information. It's clear so that I understood so how it is like going to work like that after you explained. Thanks a lot.
Yeah, and you want to do this early enough so that you have time to pivot if FDA asks you to do something differently. You don't want to be starting the clinical, you don't want to be starting whatever study, your performance testing, your animal studies, whatever. You don't want to start that testing and then submit the pre-sub and then get feedback from FDA after you've already, started your study. I mean, you can do that, but the likelihood that you're going to have to now explain why you didn't incorporate FDA's feedback, it just doesn't make FDA happy. It's like with anybody. So I'll give you an example, okay, that maybe is relatable. So it was the middle of the pandemic. We're doing like outdoor dining for Valentine's Day. Okay, my wife and I, we went to a restaurant and they had a set menu. Okay, so there were like 3 main courses and then add-ons and this kind of thing, right? And so the person comes out and asks us what we want to order, and we give them our choices. Well, they go back and then come back out a couple of minutes later and tell us, no, that option is not available anymore, right? And so there were 3 options, right? So now we're down to 2. Well, we made another selection. They go back, they come out again, and they tell us, this is we only have 1 now. And they did that for the appetizer, for the main course, for the dessert, for everything. We went through this rigmarole where they didn't tell it, and maybe some of it was as other people were ordering, maybe there were 2 options and then it was just 1, but we were always that whole time we would put in a first order, then they'd go back, they'd come out, and they tell us, well, sorry, no, we only have the 2. And then they'd go back and they'd come out, well, we're really sorry, but there's only the 1 left. They did that to us for every course of the meal. And my wife still rolls her eyes about that whole experience. So it's, this is not what you want to do with FDA, right? You don't want to go to them and ask questions, have them give you feedback, and then go do whatever you were going to do anyway. Like that's just not an optimal outcome, okay? Like this is they're not going to say nice things about you behind your back. Like that's not anyway. So I don't know if that resonates at all in terms of like, you know, if you're gonna ask FDA questions, you know, you want to be able to incorporate their feedback.
I mean, I think it resonates very well. I think it also goes back to how certain companies deal with 483s or warning letters. So, for instance, Philips, which is a Dutch company, and I'm from the Netherlands originally, they, you know, I think there probably has been a fair amount of negotiation between FDA and Philips regarding some of the findings that I think did not go very well.
Yep.
And for one of the previous classes, we had to read the response from Philips to a 483. I don't think there was a warning letter yet about one of the products that are now off the market in the US. And I have no expertise in this whatsoever, but the tone of that letter is just very defensive, not very collaborative. And
Yep.
it's like a 93-page letter, and all Philips is saying is we're gonna hire more people here and we're gonna hire more people. But they don't say, you know, we're going to do this, we're going to change this, we're going to measure the effectiveness by doing that. And if it doesn't work, we can. So I just think that that doesn't help, you know. I mean, that's just, I think, because yeah, that's what I think. It's not very collaborative, and I'm sometimes wondering maybe that's why that consent decree was
Yes.
issued, and they had to take, you know, and the stock really went down. So, and then what was interesting in the Dutch newspapers, they phrased it like, well, this is a US problem, this is the FDA. And I'm thinking, well, Europeans use the same devices. I'm sure they have these issues as well.
Yep.
So, yeah, I do think, yeah, to your point, a collaborative approach is most likely going to lead to a better result for both.
Yeah, and also just being a little bit more definitive about what your plan is. So you don't want to go to FDA and say, well, what do you think we should do to fix this problem?
Yeah.
Or what do you think we should do to show that our device is safe? You should propose to FDA what you plan to do. And some of that you can, you know, you can pull up information that is available in terms of some of the 510K summaries about what other testing was done for other products. And then you come up with a testing strategy and you say, well, this is what we propose to do based on the predicate devices that we've identified. FDA, do you have any, do you agree that this is a reasonable, approach to show substantial equivalence? And FDA will tell you what they think. Pre-submissions are a bit of a double-edged sword, though, so keep this in mind, because if you go to FDA and you ask them questions, they are probably going to give you a slightly more conservative answer. There are some nuances here where that's not the case, but if you propose to them what you're going to do, they will probably push back more comfortably in a pre-submission scenario than they would in a regulatory submission. So if you give them 80 subjects in a clinical study in an actual 510K or a de novo is a little more challenging because usually there's more interaction with FDA anyway there. But let's say that you go to FDA in the 510K and you give them 80 patients in a clinical study, showing that your device is similar to the predicate. And it turns out the predicate also did like, I don't know, 85, right? Well, there are 3 other predicates that did 100 or 120, and FDA really wants to see 100. That's what really makes them comfortable. But they see you're 80 and it's in the submission and they got to make a decision and it's good enough and we're going to give you a pass. But if you come to them with a protocol proposing 80, they're going to tell you, we want to see 100. And then if you come into the submission and you only do 80 instead of 100, and you don't have a really good justification for why you were only able to collect 80 instead of 100, they're going to tell you, look, we told you we want 100, you only collected 80, go collect another 20 patients. Like you just end up in a scenario where you probably need to bring more data if you have some of these interactions with FDA before. So whereas if you're really close to the bar and you know what the bar is, sometimes you just want to submit it.
Yeah.
You don't want to ask FDA. You just want to see if you can get it in and over. And as a hedge, sometimes, you want to, maybe go collect a couple more patients under the study while it's under review by FDA. So if they come back and say, hey, we want more. And there are nuances here. You got to talk to the statisticians about whether you can do this and whether it helps and all this kind of stuff. But there are a number of scenarios you can work out for, addressing these kinds of issues where you go to FDA with less data. And in the meantime, you're maybe collecting some more data. You might be running some more biocompatibility or performance testing or extending your shelf life, et cetera. Like there are all kinds of additional tests that you might be doing after you submit your 510K or your de novo or your PMA supplement to FDA. And I generally, and usually what that is, where it might make it through, but there are some areas of weakness. But don't wait for that always, because if you're always just waiting for everything to be perfect, you're going to submit and FDA is going to ask you some new questions anyway, probably. So I generally advise people, if you can, go in with what's good enough. If you can justify it to yourself, try to justify it to FDA, and the worst they can tell you is no. And you don't generally lose anything but time, but you would have lost time anyway going and collecting those 20 patients before you submitted. So you might actually get your change onto the market faster because you started going into FDA. They come back and ask you for more. Oh, well, we've been collecting additional patients outside of the US. Here's another 20 subjects. The data also looks great. And FDA says, sure, like we're fine with that. So anyway. Well, I'm gonna wrap. If you want, feel free to jump on over to the LinkedIn thing, but don't feel obligated at all. I'm gonna just field a bunch of questions from people who aren't in the class. Any final questions or anything from anybody before I let you guys go and enjoy your holiday? Sorry to sorry to distract you here.
No, that was very informative. Thank you so much.
Good.
I have a small question if
Sure.
you have time.
Yeah.
So I was thinking, say we have a device that is similar to device A, but also has an extra feature that is similar to device B, only that feature. So can we use both the two devices as predicate devices and explain what is taken from where?
It depends. Generally, yes. And you address that as a reference device, usually. So let's say that you're using the same material as device B, then like that's pretty straightforward. You would use the indications for use and design and all of this for device A, which is your primary predicate. And then you reference the material that's identical from device B, and that might reduce the amount of testing that you have to do, for instance, because it's in the same area, it's the same material, or maybe there's an additional software feature and you just identify that this software feature is out there and available. There might even be a separate product code for that as a, I don't know, it depends. And maybe your device is software and it's integrating multiple software features. This gets complicated at some point, depending on what the classifications of each of these reference devices are. For materials, it's more straightforward. But if your device A and device B are in two different classification numbers, it gets more problematic unless it's a system. So where maybe it's not a feature, maybe it's an accessory for your device. So device B has the same accessory as you do, that might be fine. But FDA does not mix predicates. So let's say that you have the same design as device B, but you have the same intended use as device A. This is where it gets more tricky, right? The FDA doesn't let you do what's called split predicates, where you address the performance issues with one product, you address the intended use with another. You have to be able to walk through that whole substantial equivalence discussion with one device, probably device A in this scenario. And then you reference device B for this feature that's similar, just for the sake of giving FDA some of that context. But you still have to demonstrate substantial equivalence to one of those devices.
One. Yeah, thank you. That's clear.
Absolutely. Well, have a great 4th, everybody. Try not to work too hard out there.
Thank you. You too.
Thank you.
Mostly Barbara, I think, is the person that's directed at.
Bye. Have a good 4th.
Bye.
Bye.
You too.
Bye.
Bye.